I'm Guenther Deuschl. It is today the 20th of June 2016. We are here at the International Congress of the Movement Disorder Society, and I have the pleasure to interview Professor Thomas Gasser from Tübingen. Thomas, thanks that you made it. We are very happy that we can interview and hear a little bit more about your career. I would like to start with a question about your very early steps. So what was your education, where were you educated, and how did you come to medicine?
Well, thanks. It's a pleasure to be here. I was born and raised in Stuttgart in southern Germany and decided to study medicine, I think mostly because I was interested in the science behind medicine. It was really clear to me that I wanted to become an academic medicine person. And the alternative would have been biochemistry or biology or something like that. And then I decided on medicine. I started medical school actually at the University of Hohenheim, which doesn't really exist anymore as a medical faculty. At that time, Stuttgart tried to implement a medical faculty, and they started with the first year because it was a veterinary school. So I started basically the first year in a veterinary school with the first courses, but then they decided not to pursue that, and then I moved to Freiburg. And I did my most of my medical education then in Freiburg in the first years. But as I said, I was already convinced that I wanted to go into academia, and so I talked to the student dean at the time, and I said I want to go to a lab in the United States. I want to really know what science is about. And he happened to be a hepatologist, and he said, I have a good friend who runs a hepatology lab at the pharmacology department at Yale, and if you want, I can try to set something up. And I was interested in cancer research at the time, liver metabolism. That was from all the biochemistry background. And that's how I spent the year after the Physikum, so after the first 2 years of education and after the first...
So even before finishing medical school, you went to the
finishing
States.
medical school,
Okay.
I went for a year. I got a stipend from the Studienstiftung, and I went for a year to a lab and did real lab work. At that time, it was basically metabolism, liver metabolism, with a goal of improving chemotherapy treatments for liver cancer. He was an
So you were a member of the Studienstiftung, which is one of the prestigious student awards that you can get in Germany. How did you become that?
There is a selection round just around the final exams of high school, of gymnasium, and that
there
was
you made it.
proposed by the school. I think that's how
Okay.
it works.
Okay.
That's how they got in there.
Good.
And they gave me the chance to do this year in the United States, and that's when I really decided that's the right track for me. I want to do academic medicine.
Okay, wonderful. And then you came back and finished your medical school in Germany, also in Freiburg? Or
I was also in Freiburg, yes. After that, I stayed in Freiburg. And at that time, I was a little undecided on whether I should go on in the hepatology cancer field, metabolism field, or if I should go into neuroscience. For a long time, I wasn't quite decided. And then I decided after graduating from medical school to give neuroscience a shot. Then I went to the psychiatry department at the Max Planck Institute in Munich, which was partly due. I was a conscientious objector. That was a time when you still had this trial, when you didn't want to go to the army. You had a trial, and that was I was a conscientious objector, so I had to do civil service.
Okay.
But as a medical student, I could postpone my civil service until after after finishing medical school.
Okay.
And the psychiatry department in Munich was one of the few places where I could do my civil service as a already graduated physician.
Okay.
And so I basically combined the civil service and my first year in psychiatry, and at that time I also got some exposure to neuroscience already at the Max Planck Institute.
And did you do depression, which was the main focus of the Munich Psychiatry Institute, or did you go into any other field?
I went to a number of the wards. So I was on a general psychiatry ward, I was on an addiction ward, and I already realized, it's probably not psychiatry. Neuro is fine, but it's not going to be psychiatry.
Okay. Too many patients.
Well, I mean, it's just a very different style.
Okay.
All the rounds, sitting there discussing patients for hours in those rounds. That wasn't really it.
Yeah.
And in fact, what happened then, I mean, that really makes a nice story. So we were sitting in this lunch rounds where everybody was smoking a lot in those little rooms, all the teams, the therapists, the physicians, and we're talking about all the patients and about a lot of other things, actually. And there was the phone rang, and somebody said, "It's for you," and I took the phone, and that was about a year. I was a year into that time. And the person on the phone said, "My name is Ertl. You don't know me. Are you interested in Parkinson's disease?" That's when my career in Parkinson's disease started.
Oh, that's interesting.
Because he had a position to fill. He had a grant from I think at that time it was Schering, who developed Liserite as a subcutaneous infusion dopamine agonist. He had a position to fill, and my application was actually lying at the neurology department, at Thomas Brandt's neurology department already.
Okay,
and
so
apparently
he
had handed on the application. And he called me, and those were his first words. I still remember this as if it was yesterday.
Very good. So that answers a little bit already my second question, and that is how you came into your field. So that was more or less by chance that you have chosen Parkinson's disease, or was there more behind?
Well, this was certainly a little bit by chance, but I think it also was. In a way, it probably was a little bit destiny, because when I was at the Max Planck Institute, I had the opportunity to do a little bit of electrophysiologic research, and I realized I never was the type who was too much into potentials and electricity. Mm-hmm. I always played with a chemistry set as a child, and not with the electrical set. I was the wet type of
researcher.
And so I think it just came as at the right time that Wolfgang Oertel offered me that position, because I realized everything that's more in the biology, cell biology, pharmacology direction would be more suitable.
Okay. And what were the first studies in these days that you interested most, and where you were burning for?
Well, at that time, the big thing was dopamine agonists. It was continuous dopaminergic stimulation. It was Liserite pumps, pharmacologic testing, apomorphine tests. It was very, very clinical differential diagnosis, early differential diagnosis. Those were the first studies that I was involved in.
I remember that you have done a study in these early days about the apomorphine test. Mm-hmm. That was one of your major contributions in these early days. Can you tell us a little bit how you came across this?
Well, that was I think it was just after the first paper appeared on apomorphine that actually Wolfgang suggested that to me. And so we started this first study, very systematically analyzing patients and following them up in terms of their dopaminergic responsiveness. And I think it was one of the larger early studies on apomorphine.
Absolutely, yeah. It's still a highly quoted paper nowadays. Let's just look a little bit around the background in these days. Wolfgang was in these days in Munich Wolfgang Oertel. He was one of the seniors. He had his working group, and I think it was quite a large working group, wasn't it?
Yep. He was always very talented in attracting money.
Okay.
And his group was growing rapidly. Claudia Trenkwalder joined us early on. Johannes Schwarz, Andreas Kupsch a little later. But Claudia was very early. I think we were on the same ward that when we were junior residents on the same general neurology ward. And there were a lot of people from his group who actually made it in movement disorders.
That was in Germany. That was one of the foci where scientific movement disorders began. Definitely.
Yeah.
Okay, let's go a little bit further in the development. What were your next steps then?
Well, maybe the major turn that I took was then to go into the genetics field. And I have to give Wolfgang credit again for that, I have to say. I was also funny, I remember that. He said, we were sitting there talking, and he said, genetics is really a coming field, why don't you write a review about it? I don't know anything. First you do some work usually, and then you write a review. Well, Wolfgang said, you could write a review for the Nervenarzt. And so I went to Thomas Meitigan, who was at that time at the genetics department, human genetics department in Munich. And he was interested also in neurologic diseases, genetics of neurologic diseases. And I asked him if he would do it with me. And so at that time we wrote this first Nervenarzt article, which was really the first one that appeared in German language at that time, that listed the few genes that were known then at that time. I mean, even Huntington's disease gene was not known. It was just the locus that was known. And we elaborated on the indirect diagnostic testing that you could do at that time and things like that. And that was really when.
So that was when?
That was in. Must have been 1990,
1990, 93, the publication of
maybe that was 93, but then also I thought, if I and that was absolutely fascinating me. And then I realized, with apomorphine and all these things, it's nice, but that's really something where the future is for me. And that was when I decided to go for another postdoc, or go for a postdoc another time to the US and really learn about it.
And then where did you go?
And I went to Boston, to Harvard Medical School. That was in 93, 94, to Zandra Breakfield's lab. She, at that time, was just in the final stages of hunting down the dystonia gene, the DYT1 gene, with basically one graduate student and one technician. That was Laurie Ozelius in the lab, and a technician. And I went with a DFG stipend to her lab. And at that time, actually, the Zig Zolek, who was at that time still in Nebraska, he was kind of a lone wolf who was the only one who believed that the Parkinson's disease has something genetic to it. And he was collecting Parkinson's families and was looking for a geneticist or a neurogeneticist whom he could convince that there's something in it. And he couldn't find anybody because everybody said, Parkinson's disease is not a genetic disease. Leave me alone. I don't want to waste my time. And well, I didn't know anything about it, so I thought, why not? And he sent the DNA, and that's when we started analyzing those Parkinson's families in 93. And one of those families later actually was one of the ones that actually had a LRRK2 mutation. So 10 years later then we finally identified the LRRK2 mutation in a family that originally came from Zig Zolek at that time.
Okay. So and what were the topics that you mainly worked on in Boston? That was
Well,
Parkinson's?
it was a little bit dystonia, but that field was basically taken by Laurie, and I mean that was really Laurie's project.
Yeah.
And I helped a little bit in the sequencing. I mean, finding genes at that time was a very cumbersome enterprise. You know, you almost wouldn't believe how, you know, we still had radioactive gels. There wasn't even a sequencing machine. We always poured our radioactive gels in the hot room, and there was a big fuzz about all the radioactivity that you had to work with, and very, very slowly, base by base, sequencing through genes. So there was a lot to do just in terms of manual labor there, setting together those genes. And my own project was then to start looking at those Parkinson's families, and the first things we did was we excluded candidate genes. So we had the families, and there was always the thought, no, does it have to do with a dopamine transporter, or a dopamine receptor mutation, or a tyrosine hydroxylase mutation? So we looked for markers in those genes and tested if they co-segregated with the disease in those families. And so my first paper on Parkinson's genetics, which went into Annals of Neurology, was exclusion of 6 candidate genes. Now this is almost, I mean, it sounds prehistoric, that you could, without finding a gene, without even having indication, just by excluding some candidate genes, you could get a paper in Annals. But it was a very different time.
Yes, absolutely, absolutely. Well, before we go now more into your scientific merits, can you explain us a little bit what your role models have been during your early career?
Well, I mean, Wolfgang certainly was a figure that was very important to me. At that time, very early on, I had the pleasure to get to know Anita Harding, who unfortunately very early died. But she was a great person, and when I had my first poster on those genetic families, she came up to me at an American Congress, and I was very friendly and forthcoming, and involved this little postdoc into her world, and that was really quite something, and that really gave me the feeling that there was a family, who worked together. Genetics family who liked to work together, and who took me up into the family. That was a very important encounter, I think.
Yeah. Okay. Okay, that brings us to the point. I mean, what do you think are your most important contributions to the field of Parkinson's disease?
Well, maybe one important contribution that's not really tied in a paper is that early on I started to believe that genetics does have a role in Parkinson's disease, and I pursued it, and I started to publish papers on it, and that maybe eventually convinced, and many others of course did that then over time, but certainly was one of the early ones, because I do remember a lot of people who said, don't waste your time, and it's nonsense to do that. And I thought those families were quite convincing, and there must be something to it. And in terms of actual hard findings, I think the LRRK2 gene turned out to be a major thing, which was, of course, as always with those things, a lot of luck with it. And it was not just our group. Of course, it was a collaboration we had with the Mayo Clinic at that time, who contributed, and Thomas Meitinger, still the Munich geneticist. So that was our consortium. And there was another consortium with Andy Singleton and John Hardy, who then published in the same. Yeah,
and you published back to back this I think, which has been one of the most important genetic findings for Parkinson's disease ever. I mean, still now we know that among the monogenetic diseases, it's the most frequent one, and that turned out then afterwards, certainly. Yeah, I think that's common sense, that this has something to do with Tom Gasser. Any other issues that you think have been of special importance?
Well, I mean, the other thing then later when we turn to more the genetic contribution to sporadic Parkinson's, with the GWAS studies, there Andy Singleton and I led the first large GWAS study that was published in 2009. And that really established that alpha-synuclein and tau are the major genetic risk factors for sporadic Parkinson's disease. And that was, in a way, very different because it was a big consortium effort. And much of the energy in those large consortia obviously goes into motivating everybody, keeping the group together, keeping everybody happy with their contributions. Which is really part of today's science, I think. It's very much a team science, at least in genetics. That has changed a lot. When I started, it was still very secretive. And Xandra Breakfield's lab was right next door to the lab where the Huntington's disease gene was cloned. I had not heard a single word before it was actually accepted, the paper. They actually kept it so secret. And that has completely changed. The genetics community has become much more open, much more team-oriented, and that's a very good thing.
Yeah, absolutely. And where do you think that the field of genetics will move in the next years?
That is hard to say. I must say, it's still moving unbelievably fast. It's very technology-driven, and we're in the era now of exome and genome sequencing, and I don't think that we have seen even a fraction of what this will bring in terms of results, because we just had a meeting 2 days ago of the Parkinson's Genetics Consortium, and we really think that we're way underpowered to really appreciate all the rare variants, the role of the rare variants that will come up with exome and genome sequencing. We have no idea what the non-coding genome really does, or at least only a very, very tiny amount of it is known. And there must be so much information hidden in there that it's hard to say where this will really go, but I'm sure we'll be in for a lot of surprises.
But I mean, when it comes to Parkinson's disease, there are always two competing causes named, and that's the environment on the one hand and the genetics on the other side. I mean, how are you going to solve this in the future? How do you want to entangle these two things?
Yeah. I mean, you're completely right, but I think the future will be not to see those as opposing entities, but to see their interaction. Yeah. And of course, there is no gene that acts in some vacuum. Yeah. Every gene acts in an interaction with an intracellular environment, with messages going in and out of the cell. There's a whole field of epigenetics that we haven't really addressed very well at all so far, because simply the technology is not really there. We would be interested in the actual seeing genes in action in a brain. How do you do that? Yeah. IPS technology is something that may bring us a little bit in there, but then unfortunately with IPS we erase all the epigenetic marks when we reprogram the IPS. So there are a lot of technical challenges.
I mean, given the relevance that the intestinal system has, so all the macro biome and metabolism and all these things, and given the fact that Braque says everything comes more or less through the vagus nerve, how are you approaching these questions with genetic methods? Are there ideas or thoughts about this?
Yeah, well, I mean, obviously there's the idea of gene-environment interaction. You could think of it as susceptibilities. Even if some aggregate inducing agent comes through the nose or to the gut. It doesn't do so in everybody. And it doesn't behave in everybody the same way when it propagates. So there is there must be susceptibilities, there must be modifying factors. So I think looking for modifying factors is something quite important. The advantage of the
Which
of
would
the
mean that you
genetics-
combine it with epidemiology, or
epidemiology, also in experimental models. The advantage of the genetic field is I don't think that the big advantage is that it explains everything. I think the big advantage is that there we have a starting point where we can be really sure that it's true. So we are really sure that an alpha-synuclein A53T mutation does something that eventually leads to Parkinson. There's millions of things that need to be explained, but here we have a point where we're absolutely sure that that's true. And that's, in other fields, that's very uncertain.
Yeah.
So that's, I see it more as an entry point. And I'm very open in terms of what comes after that entry point. I just think here's something where we can start.
if you were now, with all you know, 20 years ago, and if you could start again, what would you do differently?
Not much, I think. I think I'm pretty happy on how it went.
Okay.
I have to say, I wouldn't see any checkpoint where I would say, I should have taken a.
You would have taken a different decision here, one of the main decisions. Okay, it's interesting to hear. And what kind of advice would you give to new starters in that field?
Well, that I think of a lot actually, because I'm a bit worried about the new starters in the field. I'm afraid that they, maybe this is just my perspective, but I'm a little afraid that they don't see the how focusing on basically on a dream can really be fulfilling, and without thinking a lot of all the formalities and how this will lead to a position or will not lead to a position. Just do what you think is a really fascinating question, and have some role model in mind where you think, I might never reach that stage, but that's something that I really would like to aspire to. And I have to say, I mean, we have a number of wonderful young people, absolutely, but I also see a segment of our young people who I feel, I'm not so sure if they really get it.
Well, that's probably always the case.
But that's what the old folks always think.
Yeah. But this has probably a little bit to do with that. What is for you an adequate work-life balance?
Yeah, I mean, I have to say it's probably, it's the work-life balance is a very subjective feeling of when it's in balance, and I think that for me, as I'm sure for you and for many of our peers, we work to an extent that many others would think, are they crazy? And it only works if what you do is really what you really want to do.
You want to do, yes.